So this video is all about how water moves through plants, so the forces that drive that movement and how they work. Just to begin with, you'll probably remember from general biology or from ecology that the main way in which water moves through plants is by capillary action. So the way that this works is that these water molecules are slightly polar, so the hydrogen atoms are slightly positive, the oxygen atoms are slightly negative. And that means that these kind of opposite forces attract between molecules, and so you get kind of a sticking together of adjacent water molecules. That forms a force called cohesion, and that's enough to kind of stick these molecules together so that whole columns of water can move as one through the xylem vessels, through the roots, through the shoots, and up into the leaves. So that's kind of the chemistry of water movement in plants, but what are the forces that drive it? Well, essentially it's water potential, and most of this video is going to be focusing on what water potential is and how we can measure it. So water will always move from an area of high water potential to an area of low water potential. And so in short, basically when plants lose water from their leaves, as they do through the stomata, through transpiration, which occurs as CO2 is diffusing in, the water potential within the leaf goes down. And that means that it drives a water potential gradient from the xylem vessels in the plant to push water then into the leaves. And as a result of these differences in water potential, we get water movement all the way from the soil, all the way up the plant and out into the atmosphere through the leaves. So we use this kind of pitchfork symbol, psi, to denote water potential. And what water potential really is a measure of the free energy of a water system. Free energy being energy that's available to do work in relation to a standard, which is pure water. So the water potential of pure water is zero. As we, for example, dissolve solutes into that water, the water potential goes down. And you'll be familiar when you've studied things like osmosis with the idea that water will then travel down that concentration gradient. So it will travel from pure water to the water in which there are solutes dissolved. So the water potential, once we've dissolved solutes, becomes lower. So water potential is measured in pressure units. So 1 atmosphere, which is equal to 1 bar, which is equal to 0.1 megapascals. So to complete the picture of the forces that drive the movement of water through plants, the flow, so the speed of movement of water, is equal to the change in water potential divided by the resistance. And there are a number of factors that can provide resistance within a plant, which we'll come on to. So we talked a little bit about how dissolving solutes in water can change the osmotic potential and change the water potential, which can drive water movement within plants. But there are a number of different factors that make up the full picture of water potential. So this is how we calculate that. This is water potential, and it's equal to the sum of all these things. The first one being osmotic potential. So osmotic potential is that potential that's derived from the dissolution of solutes into water. And it causes movement of water across, for example, semi-permeable membranes into cells. And so osmotic potential is one of the main driving forces of water movement in between cells within a plant. Osmotic potential is either zero in pure water or increasingly negative as more and more solutes get dissolved into the water. So the second factor determining water potential is pressure potential. So pressure potential can be actually positive, it can be neutral, or it can be negative. So pressure potential is actually a result of the pressure applied to water by plant parts. So for example, water that's in a turgid plant cell, the cell walls of which are pushing in on the water, that water is under positive water potential or pressure potential. If you imagine water flowing up through an open system, like being sucked through a drinking straw, the sides of that system are actually pushing in on the water as it flows. So that water is under a negative pressure potential. And so these kinds of pressure potentials play a big role in the movement of water, particularly through vessels within a plant. This third factor is matrix potential. Matrix potential is the cohesive force that binds water to other objects like soil. So that can form some resistance in the movement of water within plants. So matrix potential is always negative. And for example, it might be the kind of cohesive force that binds water to soil particles that reduces the flow of water, say, into a root cell. So the final factor is gravitational potential, and that's the force of gravity working on water. And usually gravitational potential is slightly higher as you move higher up in the plant. It's also the force that drives the drainage of water downward through the largest pores within soil. So these are two really good diagrams from your textbook. And they show the kind of different water potential values that you see in different plant tissues and in the soil and also in the air. And that shows you the different forces that drive the movement of water through plants. So, for example, in the soil, you get moist soils, you get relatively high water potentials of around negative 0.1 megapascals. But the water travels down that water potential gradient to slightly lower water potentials in the roots, again into slightly lower water potentials in the xylem vessels within the shoots, again up into the leaves, and then ultimately out into the air. But where you can see the by far the biggest difference in water potential is between the leaves and the air. So that force that drives water out of the leaves and into the air is a particularly strong one, and that's why plants are so susceptible to losing moisture to the atmosphere. But when we look at this other plant here, what you can see the difference is the relative humidity within the air. So as the relative humidity rises, that's basically the proportion of the capacity of air for water that's taken up. That difference in water potential between the leaves and the air is reduced. So the movement of water from leaves into the atmosphere is reduced. Relative humidity is strongly affected by temperature. So as temperature drops, as often occurs as we move from day to night, the capacity of air to hold water is reduced, and so relative humidity just naturally goes up. So usually at night time, in most places, we have a relative humidity of kind of 90% or higher. And so that's why plants are so much more easily able to open their stomata at night and not lose vast amounts of water because of that change in relative humidity. So the loss of water from plants to the atmosphere occurs by the evaporation of water through the stomata in the leaves and into the atmosphere in a process known as transpiration. And a large rainforest tree like this one can lose as much as a ton of water. That's about 260 gallons of water per day through transpiration. And that's kind of amazing to think that the evaporation of water from this tree is so much more than you would get from, say, just a flat body of water of the same size. But the reason for that is that the amount of surface area created by this number of leaves just creates that much more surface from which water can evaporate. And that gives you a clue as to the way in which some plants will avoid water loss in arid climates. So transpiration rates are measured in millimoles of water per metre squared of leaf surface per second. And we can use this flux diagram to calculate transpiration rates. And it's a much simpler equation than it looks. So E is transpiration rates, and that's calculated by this value, which is leaf conductance. That's kind of the propensity of the leaf to lose water. And we'll talk about some of the factors that affect that in a minute. And that's times vapor pressure deficit. So that's the difference in water vapor between inside the leaf and outside the leaf in the atmosphere. So, for example, if water vapor in the atmosphere is very low, it's very dry, humidity is low, then this value will be higher and transpiration rates will therefore be higher. So leaf conductance is the movement of water from a leaf into the atmosphere. And it's affected by three major factors. Those are boundary layer conductance, cuticular conductance, and stomatal conductance. So to explain boundary layer conductance, essentially any leaf will have kind of a thin layer of air that surrounds it, that immediately surrounds the leaf. And as water evaporates from the leaf, that layer of air becomes more humid, and it kind of forms a barrier preventing or slowing the rate of transpiration from the leaf. But the level of boundary layer conductance, so kind of the ability of water to get through that layer and into the atmosphere, is dependent on a number of factors. So large, simple leaves, for example, like this one down here, tend to hold quite a thick boundary layer. And so they're quite good at, that boundary is quite good at preventing the movement of water out of the leaf into the atmosphere. More kind of intricately shaped leaves hold less of a boundary around them, and so boundary layer conductance is slightly higher. And smaller leaves have less of a boundary layer around them, and so the highest boundary layer conductance is in very small, intricately shaped leaves like this. But there are environmental factors also that affect boundary layer conductance. So, for example, if it's very windy, that humid boundary layer of air that surrounds the leaf can get kind of blown away. And so that can increase the rate of evaporation from the leaf as well, exactly like you experience when you use a kind of a blower hand dryer in a public bathroom. So cuticular conductance is the evaporation of water literally through the surface of leaf tissue. And that's usually very, very low, particularly in plants that have a waxy cuticle or in glabrous or hairy leaves that are designed to particularly to lose very little water. And the final factor is stomatal conductance. That's usually the most important factor, and that's the evaporation of water through the stomata. So it's affected by things like the number of stomata on a leaf, the degree to which the stomata are open, and the temperature outside the leaf. So this is a really interesting table from your textbook, and it compares a number of different trees in different kinds of environments. So mesic environments, those are relatively moist environments, and xeric environments, so very dry, arid climates. And it compares things like the density of stomata on leaves, the pore size of those stomata, so how big is the opening of the stomata, and then also things like CO2 conductance, so rate of photosynthesis per leaf area essentially. And there are some really interesting comparisons. So, for example, if you look at the difference between tropical forest trees and deciduous forest trees, tropical forest trees have about double the number of stomata per unit area of leaf, and those stomata are larger. And so what that shows you is that these tropical forest trees exist in a very wet habitat where there's always abundant water. And so essentially they just maximize the number and size of those stomata in order to maximize photosynthetic rate. They're not limited by water at all. In deciduous trees, they have to be a bit more careful with their water consumption, so they have fewer stomata and the pore sizes are also smaller. In the xeric environments, so the very dry habitats, we see kind of two quite distinct strategies. So in sclerophylls, these are leaves that have very tough leaves, so thick kind of waxy leaves. They have high numbers of stomata with relatively small pore size. So the small pore size should give them quite good control over those stomata, so they can close them quickly when it's very hot and dry. But they do have a lot of stomata, and they're capable of quite rapid CO2 conductance, so they can photosynthesize quite rapidly when the conditions are right. The other strategy is that we see in succulents. So these are kind of very broad plants like cacti that store a lot of water, and they have very few stomata with a relatively low pore size. So photosynthesis rate is always relatively low, but they never stand to lose a great deal of water. So it's kind of slow and steady wins the race. So for the last couple of minutes, I just want to talk a little bit about plant anatomy. So the tissue that actually transports water in plants is known as xylem. And there are several different types of xylem tissues. In more primitive plants, like land plants, like ferns and all the way up to gymnosperms, their xylem vessels are these very thin cells called tracheids. So this is an individual cell. At the end of the cell, there's not an opening. Water actually has to diffuse from this tracheid into the tracheid above. In more recently evolved plants, we see this new type of xylem vessel known as a xylem vessel element. So you can see it's much broader and it's open at the end, so water can flow easily from one element to another. The advantages of these xylem vessel elements are pretty obvious. They're much wider, they can transport more water more quickly. But there are drawbacks. So wider vessels like this are much more susceptible to what's known as cavitation. So we know that water travels through plants essentially in columns by capillary action, but it's possible as a result of stress or very low water potentials, or as a result of freezing, that water column can actually break and you get kind of an air bubble within that column. And that process, known as cavitation, is much more likely to happen in these very wide xylem vessel elements. So another element of plant anatomy that's obviously crucial to the movement of water within plants is the stomata. So these are two really good photos showing stomata. This first photo is a kind of lateral cross-section of a single stomata. So these are the guard cells, the two guard cells. This is an accessory cell adjacent to the guard cell, and this is just a leaf epidermal cell. So this here is the outside of the leaf, the atmosphere. And this area I is the intercellular space, so basically air pockets just inside the stomata. So it's the difference in water potential between this intercellular space and the atmosphere that's going to determine, when the stomata are open, the rate of evaporation through this stomata, the rate of water loss. This second picture is just a microscope, an electron microscope picture of some stomata looking externally at the outside of a leaf. So we'll talk about stomata a little bit more in our next video, which is all about how plants have adapted to different environments and different water stresses.